Hi everyone, welcome back to PW International Board on the series of one shot for your biology IGCSE 0610. And today we have another most important topic, that is biological molecules. Let us recollect the information that we have gained in the last one shot. We had talked about osmosis, we had talked about diffusion, we had a discussion about active transport. Now all of these processes, how do they occur, what is their significance, we actually studied about it all in the last section. All right, in this session what are we going to study? So we will have an understanding about what are living organisms made up of, what are the different components of food, and what are the different biochemical tests that are used to test the presence of these nutrients or substances in the food. Then, after that, after covering the theory part, we will be discussing the past year questions that have been taken from past year papers as well. We will have a discussion on that too. Let's begin with our session. So, what are the list of topics that are going to be covered in today's session? We have what are the living organisms made up of, or living things made up of. Then we have water, biological molecules, key definitions, monomers, polymers, carbohydrates, disaccharides, polysaccharides, starch, cellulose, glycogen, Benedict test. So these are the list of biochemical tests. Benedict test, then you have got here the iodine test, ethanol emulsion, then you have biurate test, and then we will also talk about the protein. Those of you who are studying the supplement of the syllabus will also be able to develop an understanding about the structure of DNA as well. So here we will write DNA structure as well. All right. Moving to the theory part. What are living things made up of? The bodies of all living things consist of various chemicals. And what are those chemicals? We have water, we have carbohydrates, proteins, and fats. Each of these substances plays a vital role, or you can say a very important role in the life processes. Now, what are the life processes? These are the life processes, or the list of processes that enable to sustain the life of a living organism, that make any living organism a living creature. All right. We will talk about water. Water constitutes approximately 80% of most organisms' body. And at the molecular level, we will say that a cell comprises more than 70% of the water as its volume. So here we can also write that water is the main component of a cell, and also that water is the main component, or you can say water is majorly constituting the most living organisms' bodies. All right. This cytoplasm, that is the fluid part inside the cell, contains various substances dissolved in water. So this is the feature of water, that it has dissolved substances in it. All right. The metabolism relies on water as a solvent. Now you have to consider this as a very important statement, that metabolism relies on water as a solvent. Since you will observe many questions have come directly from this statement, that you have to tell in which of the following processes the water act as a solvent, and you will be simply able to tick the right answer of that. We have chemical energy that are obtained from fats. So we use fats for the energy synthesis. We use carbohydrates for the energy synthesis and other components as well. But we will focus more on fats, carbohydrates, proteins, and water. All right. The chemical waste is carbon dioxide. So in the last chapter discussions, we had already had a discussion about the respiration process. And whenever we are talking certainly about aerobic respiration, it is a process in which the organisms uses oxygen to break down the glucose molecule to release carbon dioxide and water. So why are we calling these chemical waste carbon dioxide and water? They have come from that process. That process is aerobic respiration. All right. So carbon dioxide and water are the chemical waste over here. Done. Then energy is released. All right. Without sufficient water, cells dry out. Obviously, we have also had a discussion that the different kind of cells, either it is a plant cell or an animal cell, when placed in different kind of concentrated solutions, behave accordingly. or you can say behave different. And you have also observed that the plant cell kept in a very concentrated solution has lost more water molecules and have become plasmolized. On the other hand, when we are talking about animal cells, so animal cells when placed in very concentrated solutions usually die. And why did they die? Because the water molecules have, you can say, moved out from the cells into the solution surrounding it. So why are we saying that without sufficient water, cells die out? We have also seen in the experiments in the previous one shots as well. All right. Hence, affecting metabolism and leads to death of an organism. So basically, whenever a cell is going to die, the tissue will die ultimately, and the organ system, and hence the organism. All right. So water is the main constituent of the body, of the cell, of living organism as well. It is very crucial to sustain the life. All right. Then we will consider here that water facilitates various life processes such as digestion. All right. So here we have digestion, transports dissolved substances, aids waste removal. So there are a number of questions that are directly asking you that you have to select the uh, basically that process that includes water as a solvent. Which are those processes? We have one of those processes here, that is digestion. All right. Then transporting the dissolved substances, that is acting as a component of blood, aids in waste removal, that is a part of excretion process. Organisms need organic molecules too. And why do organisms need organic molecules? They need it for providing energy for life processes. And what are the life processes? We had already talked about that the list of processes that enables the living organism to sustain their life. All right. And energy is obtained from carbohydrates and lipids in food. So we are considering two sources of energy whenever we're talking about the components of food. We are certainly talking about the carbohydrate part and the fat part that is providing us with the energy. Then we have provide raw material for tissue growth and repair. So basically, why do organisms need the organic molecules? They have to provide energy to maintain these life processes. They have to provide raw materials for the tissue growth and repair so that they can sustain a healthy life. All right. Proteins in food molecules contains these raw materials. So these are some pointers according to the topic water. Then we have the list of processes over here. We have already discussed about those. So let me have a quick recall about all of these. We have the transportation process that includes whenever we talk about the human beings. All right. So they include heart, that is a pumping organ. They include blood vessels. Those are responsible to carry out the blood throughout the body. All right. Then we have the process of respiration in which the carbon dioxide is moved out from the lungs and oxygen is getting into the blood through the alveolar surface. All right, there is diffusion occurring in the alveoli, and this oxygen will be going into the blood where it is used to break down the carbohydrate, that is the glucose, to release energy in the form of ATP. And it also has complete combustion products that are carbon dioxide and water. All right. This is about the respiration process. Then we have excretion. So excretion is the removal of waste substances. All right. And all those substances that are present in excess quantity. So basically we can excrete water, excessive ions, excess salt. All right. Kidneys are crucial organs to perform this excretion process. We have the reproduction process in which the reproductive systems of male and females are involved. It also requires water. Then we have digestion process in which the water is acting as a solvent in the process throughout the alimentary canal. Then we have nutrition to obtain the kind of nutrition to repair and to provide energy to our body muscles, or you can say growing cells, or repair of those cells actually. So these are the list of processes that has water, and the water here plays a very crucial role in that. All right. Then we are moving forward with the biological molecules. What are these? All cells contain water, proteins, lipids, carbohydrates, salts, vitamins, or their derivatives. So all of the cells are containing water. Since we have already discussed that water is the main or crucial element of any organism, or you can say a part of the processes that are occurring in the living organism. Cells require water. They do require water. They require oxygen to carry out certain processes. Then we have salt, nutrients for cytoplasmic synthesis and energy production. So it is required for the cytoplasmic synthesis. Whenever the cell is going to divide, it requires water for that. Because we have already understood that the water is main component of the cytoplasm of the cell. So it will be required in the cytoplasm synthesis, then energy production as well. All right. Carbon is present in all biological molecules. So whenever we talk about all biological molecules, we are certainly talking about carbohydrate, fats, proteins, and we are left with, I guess, this is water. All right. So water, apart from water, we have carbohydrate, fat, and protein. All of these components contain carbon as an element. All right. So all of these have carbon. All biological molecules are organic. A biological or organic molecule is carbon based and made from living matter. All right. So this is a direct statement. Now, let me have your attention toward this statement that carbon is present in all biological molecules. Since you will observe many questions are coming directly from this statement, and in which you have to select other elements as well, but you are going to select carbon as the first element, or you can say the most important element, since it is present in all kind of food component, whether it is carbohydrate, whether it is fat, whether it is protein, all of those contain carbohydrates. So there will be a table like question in which you have to select the elements that are present in either carbohydrate, either fat, either protein. So you are going to take carbon for all of these food components. I hope this is understood by you all. Moving further, the carbon atoms forms chains or ring structures, creating macromolecules. So the carbon bound to another carbon molecule or forms the chains, form the ring structures, creating macromolecules. And what are those macromolecules? These are larger molecules made from smaller molecules. All right. Then we have, along with carbon, other essential elements include oxygen, hydrogen, and nitrogen. Since in the last slide I have just told you about that the table like question will come in your examinations, as we have observed the previous year papers. So you are going to select the correct elements that are found in carbohydrates, fat, or protein. All right. So there are other essential elements also: oxygen, hydrogen, and nitrogen. Here I just want to let you know that nitrogen is the element that will be present in proteins only. All right. And not in fat or carbohydrates. So nitrogen is found only in proteins. The carbon, hydrogen, and oxygen are present in carbohydrates, fats, and proteins as well, though the composition different differs, or you can say the ratios of these elements, you can say carbon, hydrogen, oxygen, differs in all of these food components. All right. Macromolecules such as polysaccharides, proteins, nucleic acids consist of repeating subunits called the monomers. Now, this is a very crucial statement, since you will observe that questions are coming that you have to select the monomer for the particular component of food, and you will be provided with a table. So, what are monomers? Monomers are these smaller units that join with each other to form a bigger or larger unit that is known as the polymer. All right. So, here I have the diagrammatic representation in which monomers are represented on the left hand side, and on the right hand side we have the polymers. The monomers, or you can simply say that mono means one in number, all right, or poly means many in number. So many single unit molecules joined together to form a bigger molecule, that is a polymer, and it has many monomers in it. All right. I hope this is understood. These polymers exhibit diverse properties. So all of these polymers have different properties having specific functions since the bonding differs, the number of carbon atoms, hydrogen atoms, oxygen atoms different in all of these polymers. So they will also exhibit specific functions, or you can say different functions among each other. Examples of polymers. So we have the certain examples of polymers which you need to consider. Though we have not seen any question coming from these examples section, but for the sake of our understanding, we will just go through that. Polysaccharides are the first example of polymers. Then we have polypeptides and proteins. Then we have nucleic acids. So all of these are examples of polymers. Then we have monomers that combine to each other to make a bigger unit or larger unit, that is a polymer. Monosaccharides, these are simple sugars, all right. Amino acids are also monomers. They link with each other to form the protein molecule, that is a polymer. And you have already just seen this word protein over here. So, how did this protein come into consideration? So, monomers of amino acids combine to each other to form the protein molecule. Then we have nucleotides. For instance, glucose, that is soluble, and cellulose, that is insoluble and tough. So here we have the comparison between glucose and cellulose. All right. So glucose is not so rigid in structure. You can say it is more kind of soluble in nature. Whereas on the other hand, you have the cellulose, which is tough and insoluble. So both of these are having different roles with each other. Cellulose is making the plant cell wall, that is rigid in nature, all right, whereas the glucose is serving as the energy source, all right. So both of these have distinct roles or different functions as well. Now you have to consider the statement that include hydrolysis and the condensation process. Hydrolysis is the breakdown of large complex molecules into small simple molecules by the addition of water. So by adding the water, the large complex molecules are broken down to small simple molecules. This is what we mean by hydrolysis. Talking about the condensation process, it is a process in which small simple molecules condense to build up large complex molecules by the removal of water. So you have to consider whether it is addition of water, whether it is removal of water. You are certainly talking about hydrolysis and condensation processes. What do we mean by hydrolysis? There is addition of water. What do we mean by condensation? So they have condensed, all right. So small simple molecules have condensed to build up large complex molecules by removal of water from them. I hope this is understood. Moving further, so basically this chapter is basically technical in terms. You have to remember the terminology, you have to remember the monomers, you have to remember the polymers, then you have the elements also. So they are directly fact based questions, you have to literally learn them, all right, thoroughly. So basically you have carbohydrates as the another topic. We have the definition. Carbohydrates are organic compounds composed of carbon, hydrogen, and oxygen atoms. And before that, let me make you understand this term by a simple term. Here we have that carbohydrates are simply hydrates of carbon. So you are going to suddenly recall it that what are the chemical elements that are present in carbohydrates. Since it is hydrate of carbon, so hydrate includes hydrogen and oxygen and of carbon. So we have carbon as the element, hydrates of carbon. All right. So I'm writing over here that these are hydrates of carbon. All right. They may be simple, they may be complex. This is about the carbohydrates. When we are talking about this simple kind of carbohydrates, we are talking about the monosaccharides. And what are monosaccharides? These are the type of carbohydrates that are soluble in water. All right, so they are readily soluble in water. These are the main source of energy in cells. So how do we get the energy? We are obtaining the energy from the monosaccharides. Can easily be transported by the blood. Yes, they can be easily transported by the blood. Examples are glucose. So we have already understood that glucose is the main component in our blood that will be providing us the energy. So it is acting as a source of energy in our cells as well. It can be readily or easily transported by the blood. It is readily soluble in water as well. So you can remember the monosaccharides by remembering the example, that is glucose. Then we have the fructose, which is a fruit sugar. So we have this statement over here, found in fruits. All right. We have the diagrammatic representation here of the glucose that is having a ring-like structure. All right. Or you can simply denote the glucose molecule having this ring-like structure. You can also call this as a 6-carbon ring structure. And how do we calculate it? This carbon number. So here we have first carbon, second carbon, third carbon, fourth, fifth, and the sixth carbon. So glucose molecule showing a ring structure is being represented in this diagram. All right. What are the disaccharides? Since we had talked about the monosaccharides, so disaccharides are those kind of carbohydrates that are made up of two monomer units. They are also soluble in water. Examples are maltose, made up of two glucose molecules. So this is the term that you need to remember for your examinations, that maltose are made up of, maltose have the monomers of glucose. All right. So two glucose molecules make up one maltose. Complex sugars, complex sugars that are disaccharides such as maltose are made from two simple sugars, and we have already understood that glucose is a simple sugar in the previous slides. All right, so simple sugar, and here we can write about the maltose that it is a complex sugar. All right, complex sugar that have been joined together. So by joining the two glucose molecule, we will obtain a maltose component. All right. Maltose is a complex sugar. So here we have the diagrammatic representation in which you can observe clearly that on combining these two glucose molecules, here we obtained the water. So water should be here. It should be written over here. This is the water component. All right. So we have obtained the maltose sugar, that is a disaccharide. This is a disaccharide and a complex sugar made from simple sugars. And what are those simple sugars? Those are the glucose over here. All right. Glucose, the simple sugar. Moving further, we have the starch, that is a carbohydrate. Now it is a stored form of carbohydrate in the plant leaves. All right. It has amylopectin and amylose as the structural components that make up the starch. Now you can observe from this diagrammatic representation that the amylopectin is branched. So it is highly branched in structure, highly branched, whereas the amylose are long chains. All right. The amylopectin are being highly branched and the amylose being long chains. So both of these comprise, or you can say constitute, starch molecule. And what are these? Cellulose. These are the fiber-like structure that are present in the cell wall, present in cell walls in plant cells. They are tough in nature. Then these are the cellulose fibers. All right. Here we have the cellulose fibers. All right. Here we have the cellulose. The plant cell walls are composed of long interwoven components. Here we make the cellulose. Then there is this glycogen. Now glycogen is a component that is made from glucose molecules and it is stored for of carbohydrates found in animals. It is a stored form of carbohydrate, or you can say these are the simple glucose molecules that are combining to form the glycogen molecule, and these molecules are found in animal cells. All right. So, to be specific, they are found in the liver of animals. Moving further, we have the diagrammatic representation here. There is one glucose unit, and these all units combine. Since glucose is a 6 carbon ring, we are mentioning here that this hexagon over here are combining to each other to form a bigger molecule, that is glycogen. So we have the part of glycogen molecule that is being represented in the above diagram. Functions of carbohydrates. Now, why do we use carbohydrates in our food and why do we consume them? So, here are the reasons. First reason is that they provide us with the energy. So, they are acting as energy source. Carbohydrates provide energy, releasing 17 kilojoule. And what do we mean by kilojoule? That is 17 into 10 to power 3 joules. So, this is the amount of energy we are getting per gram of carbohydrate molecule. All right. Through the respiration process. And what do we mean by respiration? We have already understood about that too. So, obtaining the energy stored in the bonds of carbohydrate, that is between the carbon-carbon atoms, and obtaining the energy for doing the different processes of a living organism inside a cell. All right. Glucose is the primary carbohydrate used in respiration. So, glucose is the main component here, or you can say primary carbohydrate that is used in the respiration process. Animals dissolved In animals, dissolved glucose is transported in the blood plasma to all cells for energy production. All right. So, where does this dissolved glucose uh, type of term just popped up here? So, basically, what happened in digestion process, there is this region of small intestine where the complete digestion of all the food components is occurring. Now, these small intestines are richly supplied with blood vessels. The food components are directly diffusing into the blood. All right, one of those is glucose. Since the glucose has entered into the bloodstream, so we will say we can now understand it more clearly that in animals the dissolved glucose is transported in blood plasma to all cells for energy production. Now this blood will be transported to all of the cells present in our bodies, and all of these cells utilize this carbohydrate, that is the glucose, to obtain energy to carry out different processes. All right. These are the different foods that have carbohydrates. So basically all of the food available right now that we are having in our daily lives all contain carbohydrates. So plant energy use is met by the respiration process also. They also require carbohydrate to obtain the energy and carry out different processes. So plant use glucose for respiration. Yes, this is true. Unlike animals, plant transport sucrose, which they convert to glucose when needed. So, this is the type of carbohydrate that has been transported, or you can say that is transported in the plants whenever we are talking about the plants only. So in animals, the carbohydrates were in the form of glucose that was transported in the bloodstream, whereas in the plants there is this another form of carbohydrate, that is sucrose, which is then converted to glucose whenever we, or you can say whenever the plants require it. All right. Starch serves as the primary carbohydrate storage form in plants, and it is stored in leaves of the plant. Stored in leaves of the plant. We will also have a discussion about the starch test as well. That is the add-in test that is used to test the presence of starch in any food component. All right. Then, all right. So this is the cellular respiration representation over here. The oxygen is taken in, the carbon dioxide is released out. Since carbon dioxide is the waste product formed during the process of aerobic respiration, oxygen is utilized in the process of aerobic respiration. Then glucose is consumed here. Yes, this is true. Then we have water that is also removed during the process of aerobic respiration by the plants. Then another function of carbohydrate is that the animal carbohydrate storage. So animals store carbohydrate. Animals store carbohydrate as glycogen, that is a polysaccharide. So it is made up of many glucose molecules. Glycogen is mainly stored in liver and muscle cells. All right. So you have to remember this statement as well. So where is the glycogen stored? It is stored in the liver and the muscle cells. However, only small quantities of glycogen can be stored. Here we have the liver organ, and here we have the muscle cell representation. All right, muscle cell. Then we have a test for the presence of reducing sugar. That test is Benedict solution test. It is not used to identify non-reducing sugars. So basically you have to first convert the non-reducing sugars into the reducing sugars, and then you can perform this Benedict test. All right. So let us come back to this topic, that Benedict solution test. It tests, or you can say detect, reducing sugar. So basically it tests the presence of reducing sugars. Examples: glucose, example maltose, all right, in food. Color changes from blue to brick red, and this is to be remembered very thoroughly, since you will observe straight questions coming from this statement, that what is the color change if reducing sugars are present. So it will change from blue to brick red. All right. Then we will talk about what if there is no color change, that it is remaining blue in color, that it indicates the absence of reducing sugars. So whenever there is no reducing sugar present in the particular solution, so it will do not change the color, that will it remain blue in color. All right. So blue in absence of reducing sugars, and brick red in presence of reducing sugars. All right. Positive results of the Benedict test. The test tube on the left contained a small amount of reducing sugar, whereas the one on the right has a larger amount of reducing sugars, and that can be detected by this color change, that it is showing red color, or you can say brick red. All right. So brick red color presence or color change denotes that there is presence of reducing sugar in it. Of reducing sugar. All right. We have another test for the carbohydrate, that is the test for starch. We use iodine solution for that. All right. Add iodine solution to a sample of the food. So whatever sample you are taking to carry out this experiment, you have to add iodine solution to a sample of the food. If the starch is present, a blue-black color will develop. All right. The iodine solution is generally yellow to brown in color, and it will turn into blue-black color whenever there is presence of starch. If no starch is present, the iodine solution will remain orange brown, or you can say yellow brown. So we can simply use this data later in the questions, that it has, if no starch is present, if no starch is there, that means the color remains yellow brown. If starch is there, presence of starch, it will remain, you can say, blue black. All right, the black color shows the potato contains the starch. So this experiment has been carried out on the potato, in which the iodine solution is used to test the presence of starch, which has turned blue black in color. That means that potato has starch in it. Then we have the another food component, that is the fat, or you can simply call them as lipids. So all of these food components, foods present over here have fat in them. Butter has fat, oil has fat. So these are the different kind of fats that are present in them. Nuts are there, avocados, meats, fishes, chocolates. So all of these have fats in it. What is the composition of fat? So a fat contain three types of atoms. It has carbon, hydrogen, and oxygen. So as we have discussed earlier, that the food components, these are the biological molecules, all of these will contain carbon as an element. So we had discussed about the carbohydrate, it had carbon as an element, and we are talking about the fat here, it is also having carbon as an element. Then we have a fat molecule consists of four smaller molecules joined together. What are these smaller molecules? That is glycerol and three fatty acids that make up a fat molecule. All right, we have the fat molecule representation. So glycerol is there and fatty acids are there, or you can simply see on the right hand side we have glycerol component. This is the first smaller molecule. Then we have fatty acid chain over here. All right, this is the fatty acid chain, this is the fatty acid chain, and this is the fatty acid chain. Both of these glycerol and fatty acids are linked by the chemical bonds to make a complete fat molecule. All right. What are the properties of fat? So we have the first property, that fat are insoluble in water. So whenever you try to mix a fat with water, it will not get mixed into each other. All right. Fats are that liquid. So fat that are liquid at room temperature are called as oils. So those fats that remain liquid at room temperatures are termed as oils. We have a test for that, that is the ethanol emulsion test for detecting fat and oils. What is the procedure for that process? We have chopped the food sample. So we are going to cut the food sample into very thin slices. All right, then shake the chopped food with ethanol using, that is, an alcohol. Ethanol dissolves lipids, that is fats, even though they don't dissolve in water. So basically fat do not get dissolved in water, but fat get dissolved in ethanol. All right. Then we have, what are the observations of these tests that are carried out on the food components that have fat in it. Observations are that the first is that pour the ethanol into water. All right, so mixing it, and then again you can recollect this information that fat and water do not get mixed, since ethanol in which the fat got dissolved is now pour into the water. So what will happen next? All right, so you have a right guess about that too. We will move further. If there is a fat in the food, the fat ethanol mixture forms an emulsion. All right, and what is an emulsion? That is millions of tiny droplets when mixed with water. All right, so this will test the presence of fat in a food component. The emulsion appears white, opaque, and this closely resembles milk in appearance. If there is no fat in the food, the mixture of water and ethanol remains transparent. So, whenever there is no fat that is present, there will be no emulsion that will be forming in the process, and hence you can say that the food component do not have fat in it. All right. I hope this is understood. Then ethanol emulsion test for detecting fat and oil. So we have ethanol of fat. So basically emulsion of fat droplets in the ethanol or water mixture. A positive test result for the emulsion test. Then another food component, that is the protein. So what are the what are the basically chemical elements that are in the proteins? You can guess the first element, that is the carbon. Then you have the hydrogen, oxygen, and we had also talked about that nitrogen is present in protein component. There is also small quantity of sulfur in it. All right. Protein molecules contain carbon, hydrogen, oxygen, nitrogen, and small amounts of sulfur as well. Like polysaccharides, proteins consist of long chains of smaller molecules called the amino acids. So these amino acids behave as monomers to make a polymer, that is a protein molecule. What is an amino acid? It is a kind of substituted methane. We have the actual methane as carbon bonded to 4 hydrogen atoms, in which the methane is substituted. All right. So we have the carboxylic acid group over here. Then we have amine group over here. All right. There is also an R group. There is R group. And whenever we substitute the H in place of this R group, we get the amino acid, that is glycine. So you can also remember that the simplest amino acid is simplest amino acid is glycine. All right. These are the representation of protein molecules over here. We have the monomers. So you can simply tell that this is a monomer over here. This is a monomer over here. Another molecule, another molecule, another molecule. What are these? All of these are representing amino acids. This is the amino acids. These amino acids are linked by peptide bonds. So we have the peptide bonds in between. This is the peptide bond. This is the peptide bond. This one is also here. All right. The representation of peptide bond. All right. Moving further, next we have composition. These are approximately 20 different kind of amino acids. So we have 20 different kind of amino acids that link to form the proteins, which can be joined in any order to form a protein molecule. Here we have the representation in which these beads are representing amino acids. These amino acids are linked by peptide bonds. So that is forming a whole protein molecule. And you can also magnify this particular protein molecule, and you can have a guess regarding the amino acids that is present randomly here. These are the amino acids. All right. Leucine, serine, cysteine. Also here we have amino acid representation. So all of these beads represent different amino acids. And you can also say that this is a polypeptide chains. These amino acids are linked by peptide bonds. What is the property? So they are soluble. Some proteins are soluble in water, and one of those is hemoglobin. Now, what is the hemoglobin? It is a red pigment. It is protein in nature. It is present in red blood cells. It is helping in the transportation of oxygen throughout the body. All right. You can tell here that hemoglobin is a protein molecule, a protein molecule. It is red in color. Then you can also say found in RBC. What are those? These are red blood cells. All right. Carries oxygen in blood. They bind to oxygen molecules and carry them throughout the body. All right. Here we have the another properties. Unlike carbohydrate, proteins are not primarily used for energy. So they are not basically providing us energy. If we certainly talk about that, what is the source of energy in our food? So basically, we will talk first about the carbohydrates. Then we will talk about second, that is the fat, and the proteins are not likely the energy providing components, basically not primarily used to provide energy. Protein plays essential roles in cell growth, repair, and maintenance. So this is the important feature of the protein molecule over here. Cell membranes in cytoplasm contain significant amount of proteins, and you have also understood about the cell membrane structure in the previous one shot, that it is a phospholipid molecule, and made up of proteins and lipids. So proteins are required to make the cell membrane, to make the you can say cytoplasm. All right. Then we have proteins are crucial for antibody production. And what are the antibodies here? These are the type of bodies that act against the infections. All right, by killing the bacteria, by killing out the viruses here. So these are the defense against bacteria and viruses. And where can we find the antibody over here? So here we have these Y-like structures, these wire-like bodies. All right, it has two heavy chains, it has two light chains. All right, this is how an antibody looks. Two heavy chains, then we have two light chains. All right, this is a representation of antibody. And antibody, these are also made up of proteins. Enzymes, which are also proteins. So we have a whole chapter considering enzymes, and it is very crucial as per examination view. The next one shot will be of enzyme chapters. The enzymes, which are also proteins, act as catalysts in various biochemical reactions. And by catalyst, what do we mean? Basically, they increase the rate of chemical reactions. They hasten the chemical reaction rate. All right. This speeds up the process in which whenever the enzymes are occurring, or you can say enzyme are taking part in the chemical reaction. Functions are the sequence of amino acid determines the three-dimensional shape of a protein molecule. All right. So basically, which kind of amino acid is placed one after the other determines what kind of three-dimensional shape the protein molecule is going to have. So the statement is saying it directly. The sequence of amino acid determines the three-dimensional shape of a protein molecule. Different shape correspond to different functions, or you can say specific functions. So different shapes are for the different functions as well. They are specific in nature. All right, they are particular in nature. Enzyme shapes dictate the reactions they catalyze. So enzymes having different shapes have their complementary shapes on the enzymes, on the substrates. So how can you just understand a little bit about this statement? So enzymes have active site on it, to which the substrate having complementary, you can say complementary shapes bind to it. The enzymes then convert the substrate into products and remain unchanged throughout a chemical reaction. So enzyme shapes detect the reactions they catalyze. So basically the enzymes are specific in nature for their substrate on which they are acting upon. Alright. Antibodies shape determines their binding specificity to bacteria and viruses. And where does the bacteria and viruses bind on the antibodies? So basically here we have the antigen binding sites. And what do we mean by antigen? Antigen binding sites. These are present on pathogen. And what is a pathogen? Pathogen is it can be a virus, it can be a bacteria, it can be anything that is causing the, you can say disease in living organisms. All right. Then we have the test for protein, that is the biurate test. We have to mix the food sample with water. So first of all, we will be mixing the food sample with water, in which we are going to perform this biurate test, that is the test for the proteins. Adding dilute copper sulfate solution. All right. Gently add dilute potassium hydroxide solution. A purple color indicates the presence of protein. So you can write the conclusion over here, that blue to purple color change is seen whenever there is protein present in the food sample. The purple is indicating presence of protein, and here the blue color says that no protein is there, in absence of protein. All right. So we have the two beakers, or you can say two test tubes. On the left hand side, it shows a negative result. And why can we say that this is showing a negative result? Since it is blue in color, and we have already found that out, that in blue color, it is showing an absence of protein. And on the other hand, on the right hand side, it is showing a positive result. That means it is having protein in it. All right. Moving further, there is another test, DCPIP test for vitamin C. The materials needed to carry out this process: fresh lemon juice, to test whether this lemon juice has vitamin C in it. Then plastic syringe is required. 0.1% solution of DCPIP, that is a blue dye. So it initially stays, or you can say remains blue, whenever it is not being tested with vitamin C. Or you can also say that in absence of vitamin C, the DCPIP solution remains blue. So you can write the conclusion over here, that blue in absence of vitamin C. Then you can write about when the solution has, or you can say the food component has vitamin C in it, so it becomes colorless. In presence of vitamin C. What is the procedure? Draw up 2 centimeter cube of fresh lemon juice into the plastic syringe. Add the lemon juice drop by drop to 2 centimeter cube of 0.1% DCPIP solution in a test tube. Observe, the DCPIP will suddenly become colorless because the lemon juice had vitamin C in it. As the lemon juice is added, note down the amount of lemon juice added from the syringe. The more the amount of lemon juice is required to test the presence of vitamin C, that means it has a very less amount of vitamin C in it. So basically, if a lesser quantity of any food component is required to carry out this process or carry out this test, that means it has more quantity of vitamin C in it. Let us compare lemon juice and oranges. So it will be that you will observe the colorless change, or you can say color change from blue to colorless of the DCPA solution when you are testing it with lemon juice, and there is a slightly you can say uh, a more time which is required to obtain this test for the orange juice. So that means lemon juice has more vitamin C in it, and orange juice has less vitamin C in it. All right. I hope this is understood. Repeat the experiment with orange juice. So we have the illustration with orange juice as well. Use the same procedure with orange juice. If it takes more orange juice than lemon juice to decolorize the CPIP, it indicates that the orange juice contains less vitamin C. I hope this statement is clear to you all. Moving to the comparison. Now, this is the table that you have to remember thoroughly. That means that you have to remember this by heart. You have to remember each and every point from this table only. Compares some properties of carbohydrates, fats, and proteins. We have carbohydrates, we have fats and proteins in different columns. Talking about the elements that they contain. So you can verbally tell that carbohydrates have carbon, hydrogen, oxygen. Talking about fats, they also have carbon, hydrogen, oxygen. Talking about proteins, they have carbon, hydrogen, oxygen and nitrogen. Alright. Then smaller molecules of which they are made. So carbohydrates are made up of simple sugars. These sugars are also called as monosaccharides. Fat are made up of fatty acid and glycerol, where fatty acid are 3 in number and one glycerol molecule is there. Then you have proteins. The monomers for proteins are amino acids linked by peptide bonds. I hope this is clear to you all and getting straight into your mind that we have all studied about this and we can simply solve the questions. All right. After the table completion, we will have a discussion about the DNA structure for the students having supplement of the syllabus. All right. So moving further, the solubility in water. Sugars are soluble, polysaccharides are insoluble. Then we have fats which are obviously insoluble. Proteins, some of them are soluble and some of them are insoluble. Why organism need them? So carbohydrate and proteins, both of they act as the energy providing source. So easily available energy. Fat is also behaving as the providing the source of energy, or you can say providing simply energy. The proteins are used to make the cells, to make the antibodies, to make the enzymes, hemoglobin, and it is also used for energy whenever glucose or fat are not present in our bodies. All right. So they will simply consume proteins to carry out the process. All right. Then we have structure of DNA. Composition is that the DNA is composed of nucleotides. All right. And we will understand this one by one. All right. We have this sugar over here that has 5 carbon in it. That is a pentagon having 5 carbon. It is a deoxyribose sugar whenever we talk about the DNA. And what do we mean by DNA? What is the full form of DNA? I hope you all know the full form of DNA, that is deoxyribonucleic acid. All right. Since we use the term deoxyribo, that means we are using the sugar that is deoxyribose sugar, where we have the representation in the diagram. So this is the ribose sugar here. All right. It is linked to nitrogenous base. It is linked to phosphate group of the phosphoric acid. All right. A nucleotide. So this all diagrammatic representation represents a nucleotide in which there is representation of adenosine monophosphate. All right. We have the adenine, that is the nitrogen base over here. This is the nitrogen base. Since we have just gathered a small amount of information about the structure of DNA, we will proceed term by term for the structure of DNA. Now, the composition is each nucleotide consists of, since we have just discussed that one phosphate group is there, one deoxyribose sugar is there, and one component of the nitrogenous base, in which we have 4 kind of or 4 types of nitrogenous base. These are adenine, guanine, cytosine, and thymine. So a nucleotide can only contain any of the nitrogenous base among these 4 nitrogenous bases present. Each nucleotide consists of a 5 carbon sugar molecule, that is the deoxyribose sugar. A phosphate group is there. An organic base, adenine, represented by A, thymine, represented by T, cytosine, represented by C, and guanine, represented by G. Next we have, so since we discussed about that the full form of DNA is deoxyribonucleic acid, we are focusing on the term nucleic acid. And what is a nucleic acid made up of? It is made up of nucleotides, in which the nucleotide is further made up of nucleosides and phosphoric acid. We have discussed that what is the phosphoric acid. So basically H3PO4 provides us with the PO3, that is the phosphate group. All right. What are the nucleosides? These are nitrogenous bases combined with the sugars. All right. So whenever the nitrogenous base is attached to sugar, we will say that this is a representation of nucleoside. And whenever we combine the nucleoside with the phosphate group, this will give us the nucleotide. So here we have nitrogenous bases which can be purines and pyrimidines. See, here we have divided the nitrogenous bases into two categories on the basis of rings present in them. Purines are there, pyrimidines are there. Purines are adenine, guanine. Pyrimidines are cytosine, uracil, thymine. And what is the significance of uracil over here? So, it is a nitrogenous base present only in RNA. Present in RNA only in place of thymine. So we are going to study about the bonding of the nitrogenous base with each other, where we will also talk about that the adenine binds to thymine with two hydrogen bonds and cytosine binds with guanine with three hydrogen bonds whenever we talk about DNA. All right. And what is the different whenever we're talking about the RNA? So RNA has binding of adenine with uracil in place of thymine, and the cytosine still binds to guanine. So this is the kind of bonding in RNA. These are the hydrogen bonds. All right, I hope this is understood. And on the other hand side, we have the sugar. It can be a ribose sugar, it can be a deoxyribose sugar. So ribose sugar will be considered whenever we talk about RNA. What is the full form of RNA? So that is the ribonucleic acid. And what does the ribo term mean here? The ribose sugar is being represented in the term as ribo. So ribonucleic acid are RNAs. Ribose sugars constitute DNA. Nucleotide chains. Nucleotides are joined by phosphate groups to form a long chain. Now, we just had a discussion about what are the nucleotides. Nucleotides are nucleosides attached with phosphate groups. All right. So, nucleotides are joined by their phosphate groups to form a long chain. Now, consider the diagrammatic representation over here. We have the nitrogenous base over here. These are the nitrogenous base: adenine, cytosine, guanine, thymine. All right. Then we have the sugar. This is the deoxyribose sugar since we are talking about the DNA. And then we have this phosphate group. All right. This is the phosphate group over here. The statement was saying that nucleotides are joined by their phosphate groups to form a long chain. What is the one nucleotide here that has nitrogenous base, that has deoxyribose sugar, and that also has phosphate group? So we have the diagrammatic representation in which one nucleotide is represented. So this is the one nucleotide over here. It has the nitrogenous base, that is the guanine. It has the deoxyribose sugar, that is the you can say that is represented over here. Then we have phosphate group attached to it. Now one nucleotide, another nucleotide, another nucleotide. So you can assume that one nucleotide is there, another nucleotide is there, and another nucleotide is there. All of these are joined by phosphate groups. You can observe from the left hand side that the phosphates are linking of different nucleotides. So the statement was also saying that nucleotides are joined by their phosphate groups to form a long chain. And that's how the DNA strand is being made. All right. The sugar and phosphate molecules remain consistent throughout the chain. So, sugar and phosphate molecules are found in all of the nucleotides, whereas the nucleotide differs from other nucleotide in having the other kind of nitrogenous base. Whether it can have adenine, it can have cytosine, guanine, thymine, all right. So, I hope this is understood and clear to you all. A part of DNA molecule with 4 nucleotides. So, one nucleotide having nitrogenous base as adenine, another nucleotide having nitrogenous base as cytosine, another nucleotide having nitrogenous base as guanine, another nucleotide having nitrogenous base as the thymine. Then we have DNA is a double-stranded structure. In the previous slide, on this particular slide, we had uh, we have a discussion about the one strand of DNA. It is actually a double strand. Basically, if you see that one strand is there and another strand is there, these are the two strands of DNA. All right, they are linked by hydrogen bonds between them. One DNA strand, another DNA strand, linked by hydrogen bonds. And where does this hydrogen bond came into consideration? Because the nitrogenous base bond to the another nitrogen base with the hydrogen bonds actually. So adenine will pair up with the thymine and cytosine will pair up with the guanine nitrogen base. In this way, they will form the double, you can say double stranded structure of DNA. And you can also say that, all right, here one strand is here. This is the one strand. All right. This is the single strand. This is the other strand of DNA linked by hydrogen bonds. It is basically giving us a representation of a ladder. Now we have to twist this ladder. All right. So you will form or you will obtain a structure somewhat like, all right. This is the kind of diagrammatic representation, or you can also say that this is having this kind of representation in which these are the hydrogen bonds between the nitrogenous bases. So DNA is basically double-stranded coiled structure. All right. DNA in a chromosome consists of two strands, chain of nucleotides. One nucleotide chain. All right. We had discussed about that. This is the one nucleotide, another nucleotide forming a chain of nucleotide, and this chain of nucleotide forms the one strand or single strand of the DNA. This is the one strand over here. All right. Here we have chain of nucleotide making up the one DNA strand and another chain of nucleotide. We have nitrogenous bases. So you can write adenine here. All right. You can write adenine here binds with thymine. Cytosine here binds with guanine. Adenine with the thymine. Thymine with adenine. So this is the kind of representation. These are the these orange bands over here represent hydrogen bonds. All right. If the first chain of DNA having polarity, that is 5 dash, or you can say 5 prime to 3 prime, the other strand will have the opposite polarity, that is 3 prime to 5 prime. You can also say that DNA has two anti-parallel strands. All right. You can also say here DNA has two anti-parallel strands. These strands are held together by chemical bonds between the bases. And what are these chemical bonds? These bonds are of hydrogen. So these are hydrogen bonds. All right. Base pairing has a rule. So adenine will pair up with thymine with two hydrogen bonds only. Adenine binds with thymine with two hydrogen bonds, whereas cytosine binds with guanine with three hydrogen bonds. This is the rule that adenine will always bind to thymine. Thymine will also always bind with adenine. Cytosine will always bind to guanine and guanine will always bind to cytosine unless we are talking about RNA. In RNA, the adenine will bind to uracil, or you can also say uracil will bind to adenine only. All right. The double strand forms a twisted helix. And how did that twisted helix came? So this is the ladder having two strands and it got twisted. So you have twisted the DNA helix. Alright, this is the kind of double helical structure you will obtain. Here you have the diagrammatic representation model of the structure of DNA. You can see that these orange bands are representing one strand of DNA. Now this is the other strand over here. Alright, two strands forming a double helical structure. This is the phosphate sugar chain because one nucleotide had sugar. Which was that sugar when we talk about DNA? It was the deoxyribose sugar. It has phosphate group and it also had nitrogenous base attached. So you will say that one DNA strand has sugar phosphate bonds, or you can also say that the one strand is actually a chain of sugar phosphate molecules. All right. So this is the one strand. Let me mark it over here. This is the one strand. All right. This is the other strand. It has nitrogenous bases forming the hydrogen bonds. All right. So these are the hydrogen bonds over here. This is the hydrogen bond over here. All of these are hydrogen bonds between the nitrogenous bases. So whenever you say that adenine is binding to thymine, so this is forming a base pair. So base is binding to another base, forming a pair. A single nitrogen base is bind to another nitrogen base, forming a base pair. Adenine is there, guanine is there, thymine, cytosine. The sequence of adenine, cytosine, guanine is part of the genetic code. Alright. The drawing shows part of DNA molecule schematically. Now, since we have covered the theory part of this particular chapter, that is the biological molecules, we will now proceed with the question part, and these questions are taken from the past year papers. Now, moving to the first question over here that says, large biological molecules are made of smaller molecules. All right. So, large biological molecules are made from smaller molecules is the first statement. Which row correctly matches the large molecule with the smaller molecules? And now we have to identify. Large molecules are present in the first column. Smaller molecules are present in the second column. Cellulose has smaller molecules of glucose and not fatty acids. Incorrect. Fat has smaller molecules of glycerol and fatty acid and not amino acids. Protein has smaller molecules of amino acids. Yes, these are the monomers linked by peptide bond to form the protein molecule. Starch has the smaller molecules of glucose and not the fatty acids. We clearly have option C as our right answer. The second question is, a student tests a solution for protein, and what is the test that we use for the presence of protein? That is biuret test. All right, in which if the solution remains blue in color, that means there is absence of proteins. Absence of proteins. If the solution turned purple in color, there is presence of protein. All right, presence of protein. So we have to here find the correct option that suits well. Name of the test is biuret test and color seen with a positive result, that is the purple color. We have option D as our right answer. Then we have question number 3 that says, iodine solution is added to a food sample that contains starch. All right. What is the color of the sample of the iodine solution has been added? So iodine solution turns from yellow brown, or you can say orange brown, to blue black in color in presence of starch. So blue black color here signifies presence of starch in it, of starch. We have, what is the color of the sample after the iodine solution has been added? So it is blue black in color. All right. Moving to next question. Question number 4 says, what is the correct statement about biological molecules? Biurate reagent is used for reducing sugars. Benedict test is used to detect the presence of reducing sugars. Then glycogen is made of smaller glycerol molecules. Glycogen is made up of glucose molecules. Oils contain the chemical elements carbon, hydrogen, oxygen. Nitrogen is not there. Nitrogen is present in proteins only. Protein is made up of smaller amino acid molecules linked by peptide bond, and this statement here is the right answer. Moving to question number 5, that says aquatic plant were kept in two test tubes. Each test tube was sealed to prevent the entry or exit of gases. One test tube was kept in the light and one test tube was kept in the dark. Alright. What needs to be added to the test tubes to detect any difference in gas exchange? Alright, so this is a question from another topic, but since the options included the list of tests that we have just discussed, so I have put the question over this one shot discussion. So what needs to be added to the test tube? So first of all, students, photosynthesis is a process in which plant require carbon dioxide, water and convert it into carbohydrates, releasing oxygen. Alright. So here, since one of the test tube was kept in the light, so it will perform the photosynthesis process. Alright. One test tube was kept in the dark, it will consume up all the starch that was the stored form of glucose present in it during the course of time whenever it was placed in the dark situation. So what needs to be added to the test tube to detect any differences in the gas exchange? Since we have discussed that neither the bending solution, neither the biurate reagent, neither the iodine solution is capable of detecting the gas exchange differences. So we will simply mark that C option, that is the hydrogen carbonate indicator, is used to detect the difference in gas exchange over here. Moving to next question. A test was performed on a food substance. A positive result was shown by a color change to blue-black. Now, this is a color change that can be observed whenever we are using iodine solution. An iodine solution is used to detect the presence of starch, and it turns from yellow brown, or you can say orange brown, to blue black in color in presence of starch. What solution was used for the test? We have iodine solution, that is option C as our right answer. Question number 7 says the diagram show four molecules. C represents carbon, H represents hydrogen, O represents oxygen and N represents nitrogen. Which structure is an amino acid? So amino acid is a substituted methane. So we are going to find the substituted methane in the given options here. A is the sugar. Then B here is the substituted methane, that is the amino acid. It is going to be our right answer for this question. Question number 8 says DCPIP can be used to detect vitamin C in food. Lemon juice contains vitamin C. When lemon juice is tested with DCPIP, what are the results? So basically it starts to convert into colorless. Alright, so it changes and finishes as colorless from blue. So it starts blue but finishes colorless. Alright, when lemon juice is tested with DCPIP, what are the results? It starts blue, finishes colorless. Alright, it do not stay colorless because DCPIP is already blue in color, and we have already had a discussion about that too. So we are going to that slide in which we had talked about DCPIP solution. Alright, here we have. Alright, so blue color represents the absence of vitamin C and colorless is representing the presence of vitamin C. Now moving further with our next question. Let's see what our next question says here. Alright. Alright. Then we have question number 9 that says which reagent is used to test the presence of protein in food, and for that we have biurate test, or you can say biurate solution to perform the biurate test actually. Question number 10 here says which row shows the elements that make up a carbohydrate molecule. So during the course of this one shot, I was continuously telling you that there will be questions coming in a table form in which you have to select the elements of particular food components, and we have just one type of question over here. It is asking about to select the chemical elements that make up a carbohydrate molecule. Carbon is there, hydrogen is there, oxygen is there, and how can we remember that carbohydrates are hydrates of carbon? So hydrate is simply telling us that hydrogen is there, oxygen is there, and hydrates of carbon. So carbon will also be there, nitrogen will not be there. Carbon is a yes, hydrogen is a yes, nitrogen is a no, Oxygen is a yes. So option C here is the right answer. Large molecules are made from smaller molecules. Which row names the large molecule and small molecules it is made of? Large molecule is the cellulose. It has the smaller molecule, that is glucose. It is the right option. Fat has the small molecule, that is fatty acid and glycerol. This is incorrect over here. Glycogen is having the smaller molecule of you can say glucose, and it is a stored form of glucose in animals. It is stored in liver and in muscle cells. Starch is the food stored component in leaves of photosynthesizing plants, and it has a small molecule that is glucose and not amino acid. Option A here is the right answer. Question number 12 says, what is the color change shown by Benedict solution when heated with a reducing sugar? So Benedict solution changes to brick red. So we have blue to red color for Benedict solution. What is the color change shown by Benedict solution when heated with a reducing sugar? So Benedict solution is used to test the presence of reducing sugar, in which brick red color shows the presence of reducing sugar, and we have option B in which red color is being shown. So blue to red is the right answer. Which color does hydrogen carbonate indicator turn if the carbon dioxide concentration is increased? So originally the hydrogen carbonate indicator is, you can say, red to orange in color, and if the carbon dioxide concentration is increased, it becomes yellow in color. Alright, since we have discussed one type of question from hydrogen carbonate indicator, I have introduced another kind of question that might come in your examinations. Alright, which row identify the chemical elements of the proteins? Now what are the chemical elements in proteins? Carbon is there, hydrogen is there, oxygen is there, and nitrogen is also there. So carbon is a tick, hydrogen is a tick, oxygen is a tick, nitrogen is a tick. Option C here is our right answer. Question number 15 says the table shows some large biological molecules and some small biological molecules. Which row correctly pair large biological molecules with the small molecules used to make them? So we have the large biological molecule, that is the cellulose. It is made up of small biological molecules that are glucose. This is correct. Oil, that is, you can say, the fats that remain liquid at room temperature. So oil has the small biological molecules as fatty acid and glycerol. Amino acid is incorrect. It is used to form the protein. Glycogen is having the small biological molecules as glucose. This is correct. Then we have protein that has amino acid as the small biological molecules and not ethanol. This is incorrect. First and third. Option B here is the right answer. Question number 16 says, what are the chemical elements shown in fat? So fat also has carbon, hydrogen, oxygen, but do not have nitrogen. Carbon will be there, hydrogen will be there, nitrogen will not be there, and oxygen will be there. Option A here is the right answer. Question number 17 says, glycogen is a large molecule made from smaller molecules. And what are those smaller molecules? These are the molecules of glucose. Which smaller molecules is glycogen made from? We have option C, glucose, as our right answer. Question number 18 says, what is the color change shown by Benedict solution when heated with a reducing sugar? So, whenever it is heated with a reducing sugar, it is turning blue to red. Alright. Which row identifies chemical elements in proteins? Carbon will be there, hydrogen will be there, oxygen will be there, nitrogen will be there. Option A here is the right answer. Question number 20 says the table shows some large biological molecules, some small biological molecules. You have to pair correctly the large molecules with their small biological molecules. Cellulose is made from small biological molecules of glucose. Oil is not made of amino acid. Glycogen is made of glucose. Protein is not made up of ethanol. Now, you can relate that this question has already come in 12th variant from October-November series in year 2023, and it has also come in 22nd variant also. So answer is first and third. Option B here is the right answer. Which row has the chemical elements that are contained in fat? So you can also solve it on your own, but you can, for the sake of the right answer, you can have option that includes All right, option A. This is also a repeated question. Question number 22 says glycogen is a large molecule made from smaller molecules. Which smaller molecules is glycogen made from? It is made of glucose. So it has already come in October-November series, and now it has come in May-June series from the same year, that is 2023. All right. The diagram shows part of protein molecules. What does X represent? So protein is a large molecule that is made of smaller biological molecules. These smaller molecules are of amino acids. These amino acids are linked by peptide bonds. So X here must represent amino acid. A here is the right answer. Question number 24 says the diagram represents a protein molecule. So this is the another diagrammatic representation, but since it is the same concept that has been applied to both of these questions, but do the small circles represent here? These small circles are of amino acids. They can provide you with any diagram. It can be a triangle linked to another triangle, and they can ask you that what is these triangles is signifying here whenever we are talking about the protein molecule. They can also say that what are these square molecules that are represented in a protein molecule. They can also say that try to locate what are these beads on this structure representing protein molecule. So we are simply going to mark amino acid as our right answer. What do these small circles represent here? A, amino acid. Question number 25 says, which chemical element, or you can say which chemical can be used to show the presence of carbon dioxide gas? Since we have discussed about the other test as well, so I have just put a question of another test, that is the lime water. So lime water turns milky or cloudy whenever it is tested with carbon dioxide. So we have option D as the right answer. You can also say that all the other tests are not including any kind of test related to carbon dioxide gas. All right. Question number 26 says 4 different food were tested to find out what they contained. The results are shown in the table. Which food contained protein but not reducing sugar or starch? So it must be tested with biurate solution, then with you can say Benedict solution, and also with iodine solution. So which solution can contain protein but not the reducing sugar or starch? So it should be tested with biurate test. Purple color should be there to detect the presence of proteins in it. Benedict solution test shall remain blue in color. Iodine solution test shall remain brown in color. Since iodine test is used to test the presence of starch, the yellow brown color shows absence of starch, and blue black color shows presence of starch. So option B here suits well. Benedict solution test shall remain blue in color. Iodine solution test shall remain brown in color, and blue test, the biurate test must have turned the solution purple for the presence of proteins in it. Question number 27 here is an experiment is set up to investigate the gas exchange in aquatic plants. So aquatic plants will also perform the process of photosynthesis. Hydrogen carbonate indicated solution is used is orange at the start. So it is having some amount of carbon dioxide in it. Which color is it after the 3 hours? Since the test tube is being exposed to light, hydrogen carbonate solution is there, and aquatic plant is there. It is filled with water. It will photosynthesize. It will consume all the carbon dioxide present in the test tube. Since it was showing that hydrogen carbonate indicator solution was orange at the start, and as the concentration of carbon dioxide decreases, the solution will turn from orange to, you can say, so in the presence of carbon dioxide it turns, in the presence of carbon dioxide it turns, you can say it was already at orange, and if the levels decrease, you will say that the purple color will be shown. All right. So the purple color indicates the carbon dioxide levels have decreased. And what about the red color? The red color is shown when the carbon dioxide concentration are, you can say, normal in color, normal in concentration. Orange color signifies that the carbon dioxide concentration are bit higher in concentration. What does yellow color here signifies? That it is the carbon dioxide concentration that has increased. Moving to next question. Question number 28 here says the table shows the result of food test carried out on a fruit. So we have the test that is carried out on a fruit. What did the fruit contain? So we have the list of tests that has been performed on that test. The tests that have resulted positive will have that food component in it. So Benedict solution is used to test the presence of reducing sugar. So reducing sugar will be there since it is having a positive result. Biuret is used to test the presence of protein. So it is showing a positive result. It will be showing a presence of protein in it. Ethanol emulsion is there. It is showing a negative result. That means the fat will not be there. Iodine solution is also negative. That shows that there will be absence of starch as well. So what will be there? There will be reducing sugars. There will be proteins. Option C here is our right answer. Question number 29 says the diagram shows the bases in a section of DNA from one strand only. So we have the nitrogenous bases from DNA considering one strand only. What is the base sequence of the complementary DNA strand? So if one strand has adenine, let me write it here, adenine, thymine, cytosine, adenine, thymine, guanine, the other strand will have complementary bases. Adenine will bind to thymine, thymine will bind to adenine, cytosine will bind to guanine, adenine will bind to thymine, thymine will bind to adenine, guanine will bind to cytosine. So this is the way we solve these kind of questions. If A is there, that it will bind to T. C is there, then it will bind to G. T binds to A, binds to A. C binds to G. A binds to T. G binds to C. T binds to A, and C binds to G. I hope this is understood. Option D here is the right answer. Question number 30 here says what is defined as the length of DNA that codes for a protein? And this is the statement of a gene. That a gene is a segment of DNA, or you can say is defined as the length of DNA that codes for a protein. Option D here is our right answer. Then we have what is the role of water in digestion? So it is acting as a solvent. Option B here is our right answer. Question number 32 says which statement is correct? Chromosomes are made of DNA. Yes, chromosomes are made up of DNA. These chromosomes can be visible during the cell division process. Chromosomes make up a gene. Incorrect. Each chromosome is a length of DNA that codes for a protein. This is about a gene. Different forms of chromosome are called alleles. No, this is also incorrect. Question 33 says the structure of DNA involves two strands. All right, coiled together to form a double helix. Which pairing of bases between the two strands is correct? So adenine binds to thymine. This is correct. Rest of all of these are incorrect. What describes the structure of DNA? Each strand contains chemicals called fatty acids. They have nucleotides, nucleic acids. Each strand contains a sequence of amino acids. Each strand contains mRNA. Two strands coiled to form a double helix, and this option here is the right answer. By this, we end our session here. I hope you understood each and every point from this particular session. For further sessions like this, stay connected with us. Thank you so much.